Hi there. Today we're going to talk a little bit about the structure of DNA and DNA replication. So, there's been a long history of DNA. Humans have been curious about how heredity and information in DNA has been passed down from generation to generation. So, long history of DNA. But it wasn't until Watson and Crick kind of put all the pieces together that we figured out the structure of the DNA, and from that we were then able to figure out how DNA functions. But first, let's take a quick look at the structure of the DNA molecule. So DNA is a nucleic acid. DNA and RNA are both nucleic acids, and we talked about nucleic acids earlier in the year. A couple of differences between DNA and RNA. DNA tends to be a double-stranded molecule, while RNA is a single-stranded molecule. And DNA is usually much, much longer, so an entire chromosome, while RNA is just a small section made from the DNA. And the sugar is different. We've got ribose in RNA, while we've got deoxyribose in DNA. Deoxyribose is missing an oxygen in carbon number 2. So over here you can see there's just a hydrogen while ribose has an oxygen. Deoxy means without oxygen, so deoxyribose is a ribose without an oxygen on carbon number 2 right here. So that is the main difference between the sugars. And then we've got a base that's different. So both DNA and RNA are going to have cytosine, guanine, and adenine as nitrogenous bases attached onto that nucleotide. But DNA is going to have thymine as its fourth base, while RNA has uracil as its fourth base. So just a few differences between DNA and RNA. When we're looking at DNA and DNA replication, we're going to be focusing more on the DNA nucleotides than we are on the RNA nucleotides. But there are those differences between RNA and DNA. So looking at the nitrogenous bases in nucleic acids, we've got pyrimidines that have these single ringed structures, and we've got purines which have these double ring structures. So purines, they're going to be a little bit bigger than pyrimidines. So we've got big bases and little bases. So cytosine, thymine, and uracil are pyrimidines, and adenine and guanine are purines. When we look at the base pair rule, that adenine always bonds to thymine and guanine always bonds to cytosine. There's a couple reasons why we have that base pair rule. When we look at adenine and thymine, that's going to make adenine and thymine together will make 2 hydrogen bonds, and guanine and cytosine are going to make 3 hydrogen bonds. And Chargaff, a scientist, noticed that when looking at all kinds of different species, he noticed that there's always equal amounts of A's and T's in every species and equal amounts of G's and C's in every species. And that's one clue that Watson and Crick used to kind of figure out the base pair rule. But when you look at adenine and thymine and you look at how they connect, they're forming hydrogen bonds between them. And a hydrogen bond, if you think back to the beginning of the year when we talked about water and properties of water and the polarity and polar nature of it, we talked about how oxygen is more electronegative than hydrogen. Nitrogen is also more electronegative than hydrogen. So nitrogen will also hog the electrons, making the nitrogen more negatively charged, leaving the hydrogen with a slight positive charge. Oxygen also slight negative charge, and so that allows a hydrogen bond to form between the hydrogen and the oxygen. You can also form another hydrogen bond here between this nitrogen and hydrogen, and so adenine and thymine will pair up so you can actually form hydrogen bonds between them. Same thing kind of happens in the guanine and the cytosine. We've got a hydrogen and an oxygen forming a hydrogen bond there. We've got a hydrogen and a nitrogen forming a hydrogen bond there, and a third hydrogen bond down here between the oxygen and the hydrogen. So the hydrogen bonds match up between A and D, and they match up between G and C, and so that allows them to hydrogen bond together. If we try and swap them, it doesn't match up. So if we tried to bond adenine to cytosine, here you'd have a hydrogen facing a hydrogen. That's not going to hydrogen bond. And then you'd have a nitrogen facing a nitrogen. That's not going to hydrogen bond. And then this oxygen has nothing to bond to. And so cytosine and adenine, they just can't hydrogen bond together. And if we try and do thymine and guanine, here you've got an oxygen facing an oxygen, you've got a hydrogen facing a hydrogen. Those are not going to hydrogen bond. You might get one down here, an oxygen facing a hydrogen, but that's just not very strong. So thymine and guanine also don't really match up either. So you're just not going to get the bonding the opposite way. So adenine has to bond to thymine, guanine has to bond to cytosine. So we've got adenine and guanine. Again, those are our purines, and those are going to bond to the thymine and the cytosine, which is our pyrimidines. So the hydrogen bonds have to match up. The other thing, Rosalind Franklin's picture, photo 51, is another thing that Watson and Crick used to figure out the structure of the DNA molecule. The pattern showed them that it's a double helix. The other piece of information that they got from this diagram is how wide the DNA molecule is. And knowing the width, they can then figure out which bases would fit into that particular width. If you had two purines, two big bases, it's simply too wide for that picture. If you've got two pyrimidines, two little ones, it's too narrow. So what they figured out was if you had a purine and a pyrimidine, that fits just right. So kind of like Goldilocks, it needs to fit just right. So this is too wide, this is too narrow, these two fit just right based on Rosalind Franklin's picture. So Rosalind Franklin's photo 51 was a significant key to figuring out the structure of the DNA molecule for Watson and Crick. Once they figured out how the base pair rule fits together with Chargaff's rule and Rosalind Franklin's picture, they kind of put all these pieces together to figure out the structure of the DNA molecule. The other thing that they figured out was that the DNA molecule is anti-parallel. What that means is there's directionality to the molecule. If you look at the oxygens in the sugar here, they're all facing up on this left side, but they're all facing down on the opposite side. So the two strands run in opposite directions. It's kind of like the highway. One lane goes north-south, the other lane goes south-north. So two different directions to that road. So this molecule has two different directions. So you do need to know which direction you're looking at for each of the different strands. So this side on the left goes 5' to 3'. The other strand on the right goes 3' to 5'. So again, Chargaff was the one who figured out that A's and T's are always in equal amounts and C's and G's are always in equal amounts. So that led to the base pair rule. So adenine always bonds to thymine and vice versa. Cytosine always bonds to guanine and vice versa. So look at the directionality of the molecule, how you can figure out the end of the molecule and which direction you're looking at. You kind of need to look at the sugar. And so if we look at the sugar, and because organic molecules are drawn without the carbons in the corners, every point actually represents a carbon. So we can number those carbons in our sugar. So if we start at the oxygen and we go around the molecule clockwise, carbon number 1 is over here, number 2, carbon number 3, carbon number 4, and then sticking up from carbon number 4 is carbon number 5, which that phosphate group is attached onto carbon number 5. The nitrogenous base is going to be attached onto carbon number 1. But then if we look at the DNA strand, over here on the left, carbon number 5, the top of that molecule is closest to carbon number 5, and that's what makes that the 5' end. Down here, this end is closest to carbon number 3, which makes that the 3' end. On the other strand, on the other side, this top part here is closest to carbon number 3, which makes that the 3' end. And then down here, this end is closest to carbon number 5, which makes that the 5' end. So this strand on the left runs 5' to 3'. This strand on the right runs 3' to 5'. So the DNA molecule is anti-parallel. Okay, so that is the structure of the DNA molecule. So now that we've talked about the structure of the DNA molecule and we understand the base pair rule and how the DNA molecule is anti-parallel, we can talk about DNA replication. And DNA replication is when the DNA makes a copy of itself. And it's going to do this before the cell divides. So either before mitosis or before meiosis, the DNA molecule needs to replicate. And so when it does that, you're taking that DNA molecule and you're breaking the hydrogen bonds that are holding it together. So we can then use that parent molecule, separate the two strands, and then use the base pair rule to build a new strand off of that template strand. And we can get two new molecules from that original molecule, and they're going to be identical to each other. They're going to be identical to the original parent molecule. And this process is said to be semi-conservative. So semi-conservative replication because you're keeping half old and you're building half new. So when we look at DNA replication, a quick overview, we've got the DNA molecule unzipping. We've got the old strand used as a template to build the new strand using the template strand as a guide, and you're going to add new DNA nucleotides one at a time to build the new strand. But because the DNA molecule is anti-parallel, we're going to have a leading strand, and that's the one that gets built continuously, and we're going to have a lagging strand, and that's going to get built in pieces, which will then get bonded together. Those pieces are called Okazaki fragments. And each step along the way is helped by enzymes. So if we take a quick look at that, the process in prokaryotes and eukaryotes is very similar, with a few exceptions. When we look at prokaryotic cells, because they have a circular piece of DNA, they are going to have a single origin of replication. They're going to have a replication bubble where the DNA is unzipped and moving along in both directions. So unzipping in both directions, it's going to create that replication bubble, which will continue to grow and grow and grow until it gets all the way around to the other end. And you've got two new DNA molecules made from that original DNA molecule. So prokaryotes are going to have a single origin of replication, but eukaryotic cells, because our chromosomes are linear and very, very long, we have multiple origins of replication along the way, and each origin of replication, you're going to have a bubble or a replication bubble opening up with replication forks moving in both directions. And then all those bubbles will keep growing and growing and growing until they eventually merge together. And then we've got DNA ligase enzyme that will bond all of those pieces together to create two new daughter DNA molecules that are identical to the original molecule. So if we look at some of the enzymes involved in DNA replication, again, we've got that origin of replication that opens up and unzips the molecule, and it's going to create replication forks in both directions. The helicase enzyme is going to be the enzyme that will untwist the double helix and it will separate, breaking the hydrogen bonds between the two strands. And so it's said to unzip the DNA. The single-stranded binding protein and the topoisomerase enzyme, those are going to kind of help the helicase to unzip the DNA molecule. So the single-stranded binding proteins will bind to and stabilize the DNA, and it prevents it from connecting back together. So if we look at this diagram, you can see the single-stranded binding proteins here and the helicase here unzipping. So as the helicase unzips it, it prevents it from connecting back together. So it would hydrogen bond back if it were allowed to come into contact with each other. So the single-stranded binding protein is needed to keep the two strands apart. And then the topoisomerase here is going to stabilize it ahead of the helicase. And so that allows the helicase to unzip the DNA molecule. The DNA polymerase enzyme is going to be the enzyme that will then add the new DNA molecules, or new DNA nucleotides, I should say, to build the new DNA molecule. But the problem is the DNA polymerase has some limitations. It doesn't know how to start building a new DNA molecule, so it requires a molecule to already be there so it can add the new nucleotide onto it. So there's going to be a primer that has to be added on first so that the DNA polymerase knows where to start. So kind of like when you're running a race, you need to draw a starting line in the sand so we all know where to start running from. That's what the primase does. The primase is going to add a primer on first so that the DNA polymerase can build onto. But the primase is going to add RNA nucleotides as the primer, so a short section of RNA nucleotides that will serve as a place for the DNA polymerase to start building from. So the primase is going to add a primer, and then the DNA polymerase will build from the primer on the new DNA molecules. The primase adds RNA nucleotides, which will have to then get replaced later by another DNA polymerase. So those RNA nucleotides will get removed and replaced later on. So after the DNA polymerase builds the new DNA molecule. So another polymerase goes back and changes those. When the nucleotide gets added on, so as the DNA polymerase is adding a nucleotide one at a time, it's actually adding not free-floating nucleotides, but free-floating nucleoside triphosphates. So if you look at this diagram here, here's your new DNA strand being built, and then here's a new nucleotide coming in. But if you look closely at that, it's not a nucleotide, it's a nucleoside triphosphate. So it's kind of a nucleotide, but it's got 3 phosphates. So the DNA polymerase not only needs to bind or covalently bond this phosphate onto the 3' end here, it also has to remove those extra phosphates. So the DNA polymerase, so here's a nucleoside triphosphate coming in. The DNA polymerase will cut off 2 phosphates and it will covalently bond this phosphate onto the 3' end of that growing strand. And because it can only add a new nucleotide onto that 3' end, it only knows how to attach that phosphate onto the 3' end. You're always building a new molecule 5' to 3'. So it's using this template strand. The template strand is 3' to 5'. The new strand is being built 5' to 3'. And because the DNA is anti-parallel, that's what leads to the leading strand and the lagging strand. So here's just a picture of that triphosphate. And we talked about triphosphates earlier in the year when we talked about ATP. So ATP is adenosine triphosphate. We've got all kinds of triphosphates. We've got ATP, TTP, CTP, GTP. All of those are triphosphates that can be used to build DNA molecules. So it can be used for energy, but it also can be used to build new DNA molecules. Okay, so again, because the DNA is anti-parallel and because the DNA polymerase only knows how to build a new molecule 5' to 3', you're going to end up with a leading strand and a lagging strand. So the leading strand is super easy. You put the primer on with the primase, and then you have the DNA polymerase adding new nucleotides one at a time continuously as the DNA unzips towards the replication fork. As the helicase is unzipping it, that leading strand is being built towards that replication fork. But the other side of the molecule is anti-parallel, and because of that, the DNA polymerase has to make the new strand in the opposite direction of the replication fork, but it has to move in the overall direction of the replication fork. So it's going to be making it in small sections as it goes, as the replication fork keeps growing and growing. And so the lagging strand is going to be made in a series of segments, and those segments are called Okazaki fragments. Then, because we've got all these fragments, we've got an enzyme called the DNA ligase enzyme that will connect together all of the Okazaki fragments, and all the fragments that need to be connected together, the DNA ligase enzyme will covalently bond them together. And the primase enzyme that puts the RNA primers, those primers will get replaced by DNA nucleotides, and then again, the DNA ligase will connect those pieces together. Okay, so if we look at the leading strand versus the lagging strand, let's take a look at, where is it? This diagram here. We've got the top strand here is going to be your leading strand. So the red is your primer. And then your DNA polymerase will add one nucleotide after another continuously as the molecule unzips. So the replication fork is going to the left, and so the leading strand is being made continuously to the left. The lagging strand, however, has to be made in the opposite direction. So it has to be made this way, but the overall direction has to go to the left. So it's making it to the right, but it's moving to the left. So it's making this first strand here to the right, and then it's jumping over here, making this second strand to the right, then it's jumping over here, making the next strand to the right, and so on, and so on, and so on. So all of these pieces are being made to the left, but the overall direction, all these pieces are being made to the right, but the overall direction is to the left towards that replication fork. So you've got one strand being made continuously, the other strand being made in sections. So a little bit here, 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 being made in sections. Those sections will then get connected together by the DNA ligase later. So the primer gets replaced by DNA nucleotides, and then the ligase connects all of these pieces together. And so because the DNA is anti-parallel, and because the DNA polymerase can only build a new molecule in one direction, you get a leading strand and a lagging strand. And then, of course, we all make mistakes. So we've got an enzyme that will go back and look for mistakes. So nucleotide excision repair is a nuclease enzyme cutting out and replacing the damaged stretch of DNA. So here you can see a mismatched base pair. So the nuclease is looking for those mistakes. It will cut them out. The DNA polymerase will bind on a new strand, and then the DNA ligase will connect them together. So the nuclease enzyme is looking for mistakes and fixing them. It doesn't always catch every single mistake, but it does catch a lot of them. Any mistakes that it might miss, those would then be mutations, which may or may not have an effect on the organism. Okay, that is DNA structure and replication in a nutshell, and I hope this was informative for you. And next time we'll talk about protein synthesis.